Hello, my name is Guilherme Nadur Silva, and I'll be presenting my group study, which is called Investigation of Snowflake Formation, Identifies Bacteria in Duluth, Minnesota Snow. As a matter of introduction, I would like to show this picture here uh, in our left, in which portrays the snowflake formation. And for that, we need a nucleated particle, which can be, but is not limited to bacteria. And that's why the snowflake formation and snow brought our attention because we could possibly find some bacteria on the snow here in the area. And basically, we have two types of snow here. The first of them is lake effect snow. The other one is regular snow. We are more concerned about regular snow because there is wind interference. So basically, regular snow is the interaction with the force in air and with the features in the land. And to go in depth on it, we needed to figure out where those winds were coming from. And through these windrows here, which is from the 2023 winter, it was defined what we predicted that most of our wind comes from the northwest direction. Now, talking a little bit about our methods, in order to conduct this study, we gathered snow samples from three different snowstorms here from Duluth. And after they liquefied, we could use a device which is called aeropress, in which we could separate the water part of the snow sample um, and the solid part of it, which was caught in the filter of this device. After we had this filter, we could bring this filter to a whole process and successfully isolate the DNA that might be contained on that filter. We also had impurities, regular dust, but we were interested on isolating the DNA of the sample. And for that, we used a series of buffers and reactants. The first of them was inhibitor X, which is a buffer that we use it to break the cells and release their content. We also use it extensively centrifuging techniques. Uh, and also we used proteinase K, which is a reactant that digested the proteins that were undesirable on our sample. We used AW1, AW2 buffers, ethanol, and ATE buffer to wash our sample and successfully separate the DNA that it contained. Now talking a little bit about our results, we basically performed two PCR tests. The first of them was aiming for 16S bacterial rRNA, and the second one was aiming for the INAQ gene of Pseudomonas syringae, which is a species, a bacterial species that is present regularly on snow. And our first PCR result came after we ran the gel through the process of gel electrophoresis. Our expected result was to find PCR products on the 1 kilobase band, which is on this region here. And unfortunately, we didn't find any. We found some PCR results on the top part of it, which are not conclusive because we should have them here on the 1 kilobase level. However, for the second PCR test, which also was ran through gel electrophoresis and furtherly imaged, we were expecting to find some PCR products which would indicate INAQ gene of Pseudomonas syringae on the 2.4 kilobase level, which would be approximately here. We didn't find, but at this time we found those PCR products here for one of our lanes, and for both of the tests, those three first lanes here, I'll circle both of them, they indicate the lanes in which our samples were inserted in the gel for PCR test. So they are the most important ones. And also we have the positive control here and the ladder to estimate where in kilobases is our product. And for this one here, we found results close to 0.5 kilobase, which wasn't the expected because the expected was 2.4, as I just said. However, for this PCR test here, we can conclude that possibly we have a kind of bacteria over there that has the INQ gene, which is really important and already used on the industry of artificial snow to make artificial snow, because it has the potential for being used as an ice nucleation uh, particle if the bacteria has this kind of gene. And as a matter of conclusion, both of our PCR results showed negative results for what we initially predicted. However, for our second PCR result, which is this one here, and I just discussed it, we could bring this sample that we found here on this at this level of the gel and at this length for further research using Illumina sequencing. Basically, there are two methods of sequencing DNA, which the first of them is Sanger, the other one is Illumina. For a quicker result, we would seek for Illumina. And Illumina sequencing would give us the DNA sequence, the correct one, and a specific one, which we could then search and successfully find the bacteria that was present here, and that is possibly present on a lot of our snow samples here in the region. And then these bacteria could be applied on different areas, could have possible useful information about them, which would be really interesting for our region. Here I have my references for this study, and I hope you like it and found this study interesting too. Bye bye.